So I'm going to talk about a project funded by FORMAS. It started last year. So it's actually, I don't have much results from this project, but I will tell you about the background to it and also what we have done so far and so on. We can discuss afterwards the phosphorus problem, because I'm sure you're all aware of that we are heading towards peak phosphorus. The phosphorus ores will end. Maybe it's difficult to say exactly when, but in the future there will be less phosphorus available for agriculture, and it will be more expensive, and it will be more contaminated by cadmium. So we have a lot of problems with phosphorus in the future. One part of the solution is, of course, to recycle phosphorus better in our societies. We need to either take the sludge from the sewage plants or maybe purify phosphorus out from the sludge to recycle it to the agricultural fields. But this project is actually not about that so much. It's more about, there is a lot of phosphorus in the soil already. Since we have fertilized with phosphorus for decennia, there is a lot of phosphorus accumulating in the soil, but most of that is actually unavailable to plants, at least to the crop plants we have today. But it's causing a lot of problems too, because it's slowly leaching out and contaminating our waters and so on. And the amounts are huge. I mean, there are tons of phosphorus in agricultural fields around the world. And that is enough to cultivate this land for hundreds of years. But most of it is, as I said, hardly bound to aluminium and iron in the soil. So the question is, can we get access to this phosphorus in future agricultural systems? So I'm going to take it, I like to go to the natural situation, natural ecosystems. How do they cope with phosphorus limitation? And this site in Northern California, the Jug Handle Ecological Staircase in Mendocino, is a very interesting place for this. I was there last winter when my son was doing a study year in Santa Cruz. So we visited this place. And you know that California, they have these huge earthquakes. And every 100,000 years, there is an enormous big earthquake. So the whole land is lifted up. You can see the staircase here. One of these steps is formed every 100,000 years around. So you have a chronosequence here of up to 300,000 years. That's a long time. I mean, in Sweden, we have only 10,000 years since the last glaciation. So this is a much longer time of ecosystem development. And you can see that it changes a lot during this chronosequence. Especially interesting in this pygmy forest that is formed after 300,000 years. I will show you some pictures how it looked. At the coastline you see that the soil is quite fertile here. You see a lot of organic matter accumulation in the soil. So this is a type of coast prairie which is quite productive with grasses and herbs and so on. And then you get into the forest, and it's the bishop pine that forms the forest close to the coastline. And there is a lot of nitrogen fixing plants present here, like the wax myrtle and the red alder. This is actually very similar to what we have in northern Sweden, where we have the upheaval of the land after the glaciation. There we also have a lot of alder. We also have buckwheat, I think it's called havtorn, as nitrogen fixing plants that are paving the road for the coniferous forest coming afterwards. So here you have the bishop pine coming when there is enough nitrogen in the soil. But then when you get to the second staircase, there are really big things are happening. And here you have the redwoods, the tallest trees on the planet, up to 100 meters tall. And you can see that they also form a very, very dense stand. You can see here they are sprouting sprouts around the main stem here. So it becomes really, really dense forest. So extremely productive. I think this is the most dense biomass places on the planet. So we go from the coastal prairie to enormous productivity in a few 100,000 years here. But then what happens, which is really weird, when you get to this third step, everything collapses, or not collapse, but the production is gone. So here we have these pygmy pines. You can also have some dwarf redwoods here. So it's not only that the species changed, also the growth is extremely poor. So the question is, what is happening here? One reason for this is that the phosphorus is gone. I mean, all the phosphorus in the primary minerals and so has been weathered out, and you have formed also a lot of bound phosphorus to aluminium and iron in the soil. So here, phosphorus is limiting the production. before it was nitrogen, but then the nitrogen builds up with the nitrogen fixing activity and so on. So the question then is, how do the plants cope with this phosphorus limitation? Can we learn something from that when we adjust our agricultural systems to phosphorus limitation? Yeah, it's not only this place that you see this phenomena. This is other places where you have a glacier retreat in New Zealand, Franz Josef Glacier. You also end into this phosphorus limited situation, and then there is also a dune system in Australia. So you can see this effect in many places when you have a long succession. And the classical graph by Walker and Sayers about phosphorus is fitting very well into this system here. In the beginning you have a lot of mineral like apatite and things like that, that is primary minerals with phosphorus. And then more and more it gets occluded, bound to different recalcitrant compounds. And at the end here it is a phosphorus limited system. And you here also see an iron mineral where you have a lot of phosphorus absorbed on the surface, and it's very difficult to release that. But some plants can do it, of course, but not so much our agricultural crops. But in natural system you have mycorrhizal symbiosis. This is my main study area in my research. So these fungi grow in symbiosis with the plants, and they are efficient especially in taking up phosphorus from the soil. In our agricultural system where we fertilize a lot, usually this symbiosis is down-regulated, so it's not well developed. I usually show this picture here just to show the effect of the mycorrhiza, because here you have a grass, you know, this fibrous root system very well distributed in the soil, and then you have an onion. An onion with these few roots, very coarse roots, can take up the same amount of nutrients as the grass, which of course is difficult to understand if you just see the roots. But if you realize that these onion roots are actually supporting a big mycelium in the soil that is taking up the nutrients, then you can understand that it's possible. So the onion is much more dependent on the mycorrhizal symbiosis than what the grass is. Yeah, here is another picture where you see fine roots with a lot of root hairs, and you can see that the mycelium is extending out into the soil here. And you see also the spores of this mycorrhizal fungus that are these little balls here that are outside. So it's very important for nutrient uptake, especially phosphorus, but it's also important for forming soil organic matter, because this mycelium is produced and dying all the time. So with time, a lot of accumulation of soil organic matter of dead hyphae is actually building the soil organic matter to a large extent. That is true both in grasslands and in forests. So this is something that we didn't realize before, that it's not only the leaves and needles, roots from the plants that build soil organic matter. The fungal mycelium is very important in this aspect. And they also help to stabilize the soil aggregates. This shows just how the mycorrhizal hyphae extend out from the depletion zones around the roots to access phosphorus further away from the roots. But you can also see that the hyphae, they really glue, there is a lot of soil particles on the hyphae, because they produce glues that are attaching the soil particles and help to form these aggregates of minerals and organic matter. Yeah, this is also showing that aggregation, which is very important for the structure of the soil, which I'm sure you are aware of. So, I haven't studied so much mycorrhizal fungi in agricultural land. This is what my new project will do. I will tell you more about that. But I have studied a little bit in grasslands, and this grassland is from Iceland. This is the 4-hot geothermal heated grassland. So you have a geothermal activity in this site. So down here you can see where there is less vegetation. That's a very hot area. So you have a gradient of increasing temperature when you go downward. So here we are studying the effect of temperature on the various aspects of soil ecology. But I was looking at the mycorrhiza, and what we did was that we buried these mesh bags in the soil. So these are colonized by the fungus, and then we can quantify how much mycelium is produced over time. We also amended these mesh bags with a maize compost, which is isotopically enriched. So we can actually quantify how much carbon is going into these mesh bags, how much carbon accumulates over time. So we have both the biomass and the necromass, so both living hyphae and dead hyphae, we can say. And what we found was there was actually quite a lot of material formed during this 3-year incubation, up to 120 grams of carbon produced by this hyphae per square meter. And most of this is dead, as you see here. So the orange part of the bars are the dead hyphae. So as I said before, they produce a lot and they die, produce a lot and they die, and then the dead hyphae accumulates because it doesn't degrade as quickly as expected, you can say. So, but that's nothing about phosphorus. I have to come to phosphorus now. So we also looked at the phosphorus, and we actually looked at different elements in the bags over time. And this is of course a bit complicated because we have ingrowth of hyphae that brings in nutrients into the bags. And now we have outflow from the bags, which is the nutrients taken out by the fungus into the plant and also what is leaching out from the bag. So if you look at carbon and nitrogen, this is a balance. So the same amount that's coming in by the hyphae is going out. But for phosphorus, we see a steep decline here. So after 3 years, 35% of the phosphorus was gone from the bag. So the real figure is actually bigger because we have, as I said, an import also. So this is what we expect, that these mycorrhizal fungi are important in taking up phosphorus. But this is from organic matter, from this maize compost. So it's not really the recalcitrant form of phosphorus that I'm working on now in the new project. So now we have to move to another place. This is a very nice place in the Alps, Italian Alps, in the Aosta Valley. Here we have bedrock which is composed of some serpentinite. Yeah, I can show you this picture here. So to the left you have pure serpentinite in the ground, and to the right you have gneiss, which is more common bedrock here in Sweden. But serpentinite is very special. There are almost no phosphorus at all in the bedrock. It's also very little potassium, and there is a lot of heavy metals. So it's very difficult for plants to grow in this type of bedrock or this type of soil. But we were interested in here. So the plants that can grow in this very phosphorus poor soil, how do they do it? Can we learn something from them when we try to develop our agriculture? So we can produce or get more phosphorus efficient crops. So we're studying this in a glacier forefront. You cannot really see the glacier, but up in the valley here you have a glacier that is retreating every year. So the first site we used was 5-year-old moraine or till. So the glacier left this place 5 years ago. So this was a non-mycorrhizal control or no plant control. So we have a place there where there is no plants. And then we have, after 70 years, you have the first Larix decidua trees establishing. 190 years are a bit bigger, and after further down the valley you had forest that is growing on 3,000-year-old soil. So apparently, yeah, you can see also this part of this side of the valley there was some gneiss mixed in with the serpentinite. So you can see that there is better growth here. So it's clearly that when there is phosphorus available, both the field vegetation and the trees are growing better. So we're very interested in how do these Larix trees do to get enough phosphorus from this very poor site. So what we did here, we made similar mesh bags, but here we amended them with iron oxide that had phosphorus attached to the iron oxide, so very hardly bound to the iron oxide. And we then incubated them in the soil for up to 1 year, and we looked at how much of this phosphorus can be used. So we thought about 2 possible ways for the fungus to remove the phosphorus from the iron. One was by producing organic acids, because they can replace the phosphate on the surface by competing with the phosphate molecules. So this was actually what we thought was the most important when we started the project. Another possibility is that the fungus produces iron-reducing metabolites, so that the iron in the iron oxide is reduced from iron 3 to iron 2. That makes the iron much more soluble. So that would be a way to dissolve the whole iron oxide complex and then release the phosphorus in that way. And we did a lot of calculations here, and I just summarize here that we found that between 12 and 25% of the phosphorus was released by competition with the organic acids, but much more actually, up to 49% of the phosphorus was removed by dissolution of the iron oxide. So this was really striking results, that this fungus have the capacity to actually dissolve iron oxide in the soil. We know that these fungi produce iron-reducing substances because they have been found to do that when they interact with organic matter. These fungi normally are involved in releasing nitrogen from organic matter. And one way to get access to the nitrogen that is bound in organic matter is to oxidize the material. And then they use this iron-reducing metabolite. So we knew that they have the capacity to do it, but this result suggests that it's very important also for the phosphorus uptake, not only the nitrogen uptake. So this was the basis for the new project then, that we wanted to see if different crops had the same capacity, or we didn't think they have the same as these trees in this very extreme environment, but do they have any access at all to these sources of phosphorus? Can we then improve it if possible? So we started actually here in Alnarp at Lundstop site. Maybe you're aware of this experimental field site here outside Alnarp. And we were interested in this perennial wheat, the Kernza, because it's tested there, both when it's grown alone, and it's also grown with alfalfa mixture to have the nitrogen fixing plants. And then we compare that with the winter wheat, which is more the conventional system which is tested out there. So we have installed this equipment, and you see here now we're not only interested in the capacity of the fungus alone. We have this ingrowth course where also roots, the fine roots, can enter into the course to get access to these sources of phosphorus. Here, instead of having this glacier forefront with no plants, we have installed trenches by forcing down a tube into the soil to restrict root ingrowth, so that we have we can see how much of the phosphorus can be released by just rainwater flowing through and so on. I'm not really sure it will work with this because it's of course possible that the roots come in from underneath. So we haven't really checked that yet. We haven't harvested all the experiments. So we are a bit, we'll see how it goes with that. But you can see that there is a lot of roots that have grown into this course when we harvested them. A lot of soil attached around them like this before. So they are well colonized by roots. That's a good thing. And also when you look in the microscope, you can see there is also a lot of hyphae here, all these thin threads here, and roots coming here. So, and you can see this, we have actually taken a pure quartz sand, and then we have made a very thin layer of iron oxide around the quartz. And then it's then phosphorus absorbed on that surface. So now we haven't measured yet how much of this phosphorus has actually been removed, but that will be done maybe before Christmas or early next year. So I don't have the results, but it looks good when you see the contact at least. Okay, so the next field site is the Swedish soil fertility experiments, which started in 1957. We have 4 of these in Skåne, here in Scania, in the southern Sweden. So we have used that, and this is a crop rotation. So this year they were growing sugar beets. At first I thought it was, sugar beets doesn't form mycorrhizal symbiosis, so that's not a good thing for my project. But on the other hand, sugar beets are known to be very phosphorus-demanding plant, and they produce a lot of oxalic acid. And like this is not a sugar beet root, but this is from a carex, which form this what called dosiform roots with lots of root hairs that produce a lot of oxalic acids and citric acid in a way to get access to phosphorus. So maybe the sugar beet is actually a very good crop to compare with. What is the capacity of this crop to take up the phosphorus in this iron complex? And we can then select high and low P-level plots in these different field sites. We have just harvested these, but as I said before, no results yet. But actually, in these trench plots, we could see sometimes that the roots have come in because the mesh bags were colonized by roots even in the trench plot. So maybe the sugar beets have a great capacity to produce roots that can also grow upwards into the tubes. Yeah. Yeah, here we see just before we harvested the crop. And then the third one is related to biochar. And this is something that is a really hot topic today. And I think it's interesting. Biochar is a soil amendment that are supposed to enhance water and nutrient holding capacity. It also binds carbon in the soil, so it's a way to mitigate climate change. And it also stimulates mycorrhizal fungi, because this is an enlargement of the biochar particle. And you can see all these hollows and this structure, which is very nice for water and nutrients to attach to. And also we can easily see that the mycorrhizal hyphae likes to grow into this hollow structure, because it's a good habitat for it. And the site we're going to use here is in Kenya. This is an experiment they started 16 years ago. They have maize and soybean as a rotation crop, and they were adding this biochar in rows like this when they started. And then they found an extremely good effect of the biochar. This is some slides that was sent from Kenya. And you see to the left where they didn't amend anything, no biochar and no mineral nutrients, and there was almost no growth at all. But if you only add biochar without any nutrients, the improvement of growth was enormous. And also, of course, if you combine then biochar and fertilizer, you had a very good effect on the yield. So the question is, why does it grow so much better with biochar if you don't add any nutrients with it? I mean, you have a lot of experiments also in Europe and Sweden, and you have a very slight effect on the yield sometimes. That's a little bit better growth. Maybe it's related to the water holding capacity and so on. If you have a dry summer, maybe you have a small effect. So, but you never have this huge effect. So I think this is really interesting, and I really want to find out why you have such a good effect. And one possibility is, of course, that in this very old soil, which is very aluminium and iron rich, as soon as phosphorus is released from organic matter or something, it will immediately be attached to this iron surface and not available for the plant. But if you have biochar mixed in there, so if there is a phosphate molecule released, it can go to the biochar instead and attach there, and then the plants have access and the fungus have access to it. So that's one possible explanation to this big difference with biochar and non-biochar in the tropical regions. So yeah, and we have Thomas Ketter, which is also in SLU in Uppsala. He did a study here, which actually is 10 years after they added the biochar. And they still have a very good effect on the growth, even by biochar alone. So we're going to go, now the problem with the corona, we couldn't go there to install our equipment, but we have now sent all this equipment, all this ingrowth cores and iron oxide material, and as planned, they will be installed next year. So we are sending videos how to do it and so on. And maybe we will be able to go there before Christmas to collect it, but I'm not sure at all. So it might be that they will also harvest it for us and send us with DHL or something so that we get the material after Christmas, so that we can do the same study here. Because I think this is the most interesting site, because here you have so strong phosphorus limitation in the soil. And we also try to another thing, because we have two students which are engineers working with nanotechnology. And there is a technique called microfluidics, or whatever, I'm not really sure how to pronounce it, but it's something that was developed in medical research, where they have microchips with a very very fine structure with small channels and so on, where you can build up different gradients in nutrients and so on. And Edith Hammer in our department, she has developed this into a soil chip. So this structure can be colonized by organisms from the soil. So here she has added soil around and let the organisms come in and colonize it, and you can then add nutrients there, you can put biochar there and so on, to try to figure out on a very fine scale what's going on. So here for instance you can see small channels here in the microchip, and here is a nematode starting to colonize one of these small channels. So you can see hyphae growing in and bacteria colonizing and so on. So these two students, they are really keen on going to Kenya to do this complementary study there, but so far they have not been able to do it, and they hope still to do it in the spring next year, but we'll see how the corona will develop. So, because I think this maybe an option would be to bring soil from Kenya, from this site, and do experiments here in Sweden instead. We'll see how it goes. Yeah, so this is all I had, and these are just my group in Italy that was working with this Italian study. So I'm open for questions. Yeah?
Regarding biochar, in Sweden they often recommend that you fertilize it before using it.
Yeah.
I'm just wondering, is that necessary, or is that, in your opinion?
I think so, yes. Yes.
It is necessary.
Yeah. Because, I mean, the biochar has a nutrient binding capacity, so you can actually reduce growth because some of the nutrients in the soil will be attached to the biochar, maybe less, a bit difficult to access for the plants. So it's better to load it with nutrients because the risk that it's leaching out is not big. So I think that's a good idea. Also in Kenya, I think it's a good idea to add it both, because you saw also that the growth was much better if you added both the biochar and the nutrients.
Any more questions? Not all at once. I'm thinking about phosphorus use in Sweden overall, and adding it to the soils, and just thinking about, would it be a good idea, for instance, to try to inoculate fields instead with mycorrhizae and adding less phosphorus? Is that viable options to farmers? I'm just thinking solution-wise here, how we can, at least in Sweden, lessen the use of the phosphorus and the depletion of the mines.
Yeah, that's a good idea. Well, a good suggestion. I know in Canada they have done that. A colleague of mine, he said that they have reduced the phosphorus addition, which is necessary, otherwise the mycorrhizae won't develop. And they have found, in some cases, better yield, even if they reduce phosphorus, if they inoculate with mycorrhizae. So that there are examples of that. I must say also I'm a bit skeptical, because if you buy inoculum or mycorrhizae, you can buy a few different strands which is possible to cultivate and produce in mass numbers. These are only a few strains that you can do that with. And I more like when you have a complex community of organisms that can help. So I was working in Tunisia a lot with this, where they have a lot of erosion problems. And we did one experiment by adding inoculum of a mycorrhiza that was developed in Spain, but it disappeared. It didn't do any good at all, because it was not really adjusted to the conditions there. So then instead we added a lot of compost material, like nitrogen-rich acacia compost. And there we saw much better growth of the mycorrhiza, the indigenous mycorrhiza that is present in the soil already. So I think it's probably better to try to adjust the cultivation with less phosphorus and more organic matter, compost material, and then I think you will improve conditions for the indigenous mycorrhiza rather than putting in a new one. Maybe you can do both if you have really, if there is no inoculum at all in the soil, because then you need to add something. But it's probably better to add something from a natural soil with natural mycorrhiza than to have this bought inoculum. But I have some friends working in Africa where they try to not only have, they try to develop inoculum that is adjusted to the country or to the site where it's going to be used. So it's not impossible, but you have to be a bit careful when you buy this inoculum on the net, because it's a very special strain that you can produce in high numbers, but might not be the good one for your purpose.
Do you think in Sweden and Europe overall, just thinking about, we have newer soils than you mentioned, than say in South America and do you think that the phosphorus that is in the ground there may be heavily bound today, but still might become possible if you just feed the mycorrhizae enough, so it can help with the phosphorus subject? Do you think you can actually make that shift? Take a scenario if we couldn't get phosphorus in bags starting tomorrow. Would it be able to replenish and repair the soils enough? Is there enough phosphorus in the soil as it is, or do you need to amend it organically somehow?
I think you're talking about the agricultural land now. I think there is plenty of phosphorus, as I showed you in this picture in the beginning. But then I'm not sure. I mean, what we found in Italy with these ectomycorrhizal fungi growing in symbiosis with the larix trees, they have enormous capacity to produce acids and metabolites that are important in oxidizing material and so on. But I'm not sure that the muscular mycorrhiza, which forms symbiosis with the crops, have that ability. That's actually one of the tests we want to do. So we shouldn't think about this Italian study and that the mycorrhizal fungi can do the same thing in the agriculture land. It's not. We don't know that, but that's something that I want to test. That's why I'm doing this work. So it could be that that capacity of those fungi are not so much releasing material from very hardly bound, but maybe we know it's very efficient in taking up low concentration phosphate, and of course by doing that they can the equilibrium will go towards releasing more, so to speak. But maybe it's more that effect, that it really reaches everywhere and it can really take up phosphate in low concentration. I don't think it has so big capacity to actually attack mineral material too. But that's something that future will show. Yes, Niklas.
Is it only a monetary question to retrieve already used phosphorus? If we want to recycle it, and because, as Niklas said, if we have a lot of phosphorus in our soils, it's not an issue as long as we can make it available. But if you look at other countries that doesn't have phosphorus, they will probably need it in the future.
Yeah.
And one way, if when it's all out in the source, we will have to recycle, of course.
Yeah.
Yeah. Is it that only a monetary question, or will there be a way not when it's not available anymore, and we cannot actually retrieve it?
I think more. I'm not sure, but I think that I don't think we will run out of phosphorus completely, but maybe the yield will go down. So even if we cannot afford to add as much to produce 100% yield, because maybe you have to add much more than what the plants take up, because so much is being fixed in the soil and so on. But we could be that it's not worth to get this extra 20% yield because the phosphorus will be so expensive. So it will not be black and white. It will be more expensive to produce maximal.
Yeah, that's why if there will always be phosphorus to be able to be produced, but maybe it's not monetary valuable.
No, exactly. Yeah.
Interesting. Any other questions? Everyone's taken by shock.
Yeah, here's one.
I'm a little bit it was very fast with so many different examples. So I'm a little bit confused with the Swedish studies. You're just looking at what mycorrhiza is doing in terms of taking up phosphorus in those studies as well.
Yeah.
But the first one, Kernza, is mainly to look at what Kernza can produce.
Yeah.
It's not actually looking at phosphorus as a main thing. It's more like a side effect.
Yeah, I mean, it's like a background descriptive study in a way. So can these plants take up this source that we have at all? The second question, which I haven't touched upon yet, is how can we improve this uptake? But I mean, I need to have a baseline first to see. Maybe it's not possible at all. Maybe all this iron-bound phosphate is unavailable to all the crops we are testing. I mean, Kernza was interesting because it has such a big root system. It's more like a natural plant more than the highly bred crops that we have today. So one idea is maybe that maybe it has a higher capacity than the other crops. But I mean, the really interesting question is how can we improve the uptake if we have quite low ability here? How can we improve it?
And then it looked like normal winter wheat, not like a no-till winter wheat or something like that. So it becomes very extreme if you're talking about mycorrhiza, I guess.
Yeah. I mean, these are two. I mean, if we get an effect there, like the kernels that takes up much more than the wheat. It's difficult to say, is it because of the cultivation system or is it about the species and crops? So I am aware of this. I mean, it's difficult to disentangle out the real mechanism, but this is more like a baseline type of research, and then we have to add on more specific experiments to test mechanisms. That's a good point, yes.
Was I correct, or did I understand correctly that you said oxalic acid helps, or helps to take up phosphorus?
Yeah.
That's bound to iron.
Yeah.
Could we use that in a way, with planting plants like sugar beet, and then rotating with other,
I don't know. I mean, it's well known that oxalates have this capacity, or it's said in the literature at least. But then I also know that somebody said if it's really hard bound on iron, you have to have a very high concentration of oxalate to actually have an effect. So it's not really cleared out if it has the capacity. I think the research here will show if we can remove it. But I think your idea to have a crop that can actually mobilize phosphorus could be one way in a crop rotation system. So actually, if it is that only the trees, the mycorrhiza formed with trees, have the capacity to mobilize this phosphorus, then maybe it's an idea to have a tree crop, like an energy forest or poplar or something, short rotation poplar, to actually mobilize a lot of the phosphorus that is bound there, and then you can grow agricultural crops after that. That's one idea. If it is, like you say, maybe you need a special crop to really get moving this phosphorus that is bound. So that's a possibility, yeah.
Do you have an idea like how big an area this would help with, like if you do intercropping with trees or crops like that, can it actually affect you?
Yeah, I don't know, but I mean, like in Lundstop they have this agroforestry type of plot too, where they have trees, and we thought about that idea when we have been there. If we see this effect with the trees but not with the other crops, how long can we see this effect out in the field? So that's an interesting question, but I don't know the answer.
Well, maybe you can extract the acid from sugar beet. That's not what you want in sugar anyway, right? So
No.
you can use that and spray it on the fields or something. I don't know how that would affect the environment.
No. I think it's also when you analyze the oxalic acid concentration in the soil is usually very low. So some people have difficulty explaining how can this oxalic acid have such a big effect if it's so low concentration. But then maybe an idea is that around the hyphae where it's produced, or maybe the roots in some cases, maybe you build up a high concentration enough so that you have an effect. But then a few millimeters or maybe micrometers, I don't know, not millimeters, away, it has no effect anymore. If that's the case, maybe it's the mycelium spreading in the soil that really can, then if you add more on like the sugar beet leaves or something, maybe it doesn't get into the contact to really build up the concentration high enough. I don't know. But
do you think that this new trend of like you no dig, no till, just trying to not disturb the soil as much has a positive impact on mycorrhizae?
Yeah, I think definitely that if you disturb the soil, you will enhance some of the. I mean, you won't kill all the mycorrhiza if you dig in the soil or something like that. But if you have a lot of cultivation of the soil, some species of mycorrhiza will be benefited because they can cope with the disturbance, while other species will be less inhibited. so definitely you will influence the community. But apparently, I mean, the no-till is not a so big problem. I mean, at least for the carbon point of view, it doesn't seem that the tilling have a very strong effect on the organic matter in the soil. Compared to in US, you have a very strong effect if you stop tilling in terms of organic matter formation. And organic matter is really important for the mycorrhiza. If you have low organic matter soil, the mycorrhiza won't like it at all. So by just adding organic matter, especially nitrogen-rich material, if you just add straw or something which is very low in nitrogen, you don't have this positive effect. So I think that's probably more important to add organic matter or nitrogen-rich organic matter.
So adding like green manure to your crop rotation really helps.
Yeah.
Any final questions?
Yeah.
What do you think about these new biostimulants where you get innovators, for instance, with
Yeah, as I said before, I'm a bit skeptical, but I'm not so well acquainted with them. But for the mycorrhizal inoculum, I know I've talked to many people who are experts in mycorrhiza, and some of them are actually working with this production of these. And this guy in Africa, I think, I trust him quite well, and he said, but we need to produce inoculum in the place where it's going to be used, and we have to test many different, but I mean, most of what you can buy is this single strain that somebody managed to produce in high numbers, and those kind of things I'm always skeptical to. So you have to be careful there, but I don't want to say that everything is bad, but there is a big risk there.
So final question here. So just the takeaway, what do you think students should be focusing on if they want to be a part of this change and help to lessen the effects of depleting mines and actually working towards solutions for peat phosphorus? Where do you think work needs to be done? Yeah,
I mean, like recycling is a big thing, of course, and a lot of companies working with this. I mean, if you're going to lose the sludge, or if you can purify phosphorus from the sewage sludge, not only the sewage sludge, but in all our society to recycle. That's probably the most important, I think. But from the agricultural point of view, I mean, organic matter, the mullhalten in the soil, that's the really important thing to maintain it and work for increasing it. I mean, for many purposes, also for climate change. I think we have a great capacity to mitigate climate change by increasing the concentration of organic matter in the soil. And I think then the Kernza, for instance, will be fantastic because it will really increase. But for Kernza, the yield is so low so far, so I think we have quite a long way to go until we can replace normal wheat with Kernza. But maybe in some areas, low productive soils and eroded places which are prone to erosion, it could be a lot of benefits by trying Kernza, for instance. But yeah.
Super. So thank you very much.
Okay.